Okay, so this next PowerPoint is going to go over the last parts of chapter 9, PowerPoints B and C. And we left off PowerPoint B on muscle tone, isotonic, and isometric contraction. So that's when I'm going to pick it up. And first, the concept of muscle tone refers to the slight contraction that is skeletal muscles sustain at any given time. This is due to reflexes, spinal reflexes, which let us know that even though skeletal muscle contraction is voluntary, it doesn't always have to be voluntary. It's oftentimes taken over by reflexes, which are involuntary. The difference between cardiac and smooth muscle is that cardiac and smooth muscle could never be voluntary. We could never have voluntary contractions of cardiac or smooth muscle, whereas skeletal muscle have either voluntary contractions or involuntary when reflexes take over their contraction. So that's one of the differences. The purpose of muscle tone is to keep muscles firm and healthy and ready to respond at any given time. It also helps us to maintain posture, and it keeps muscles toned, if you will. Now, when it comes to muscle contraction, there are two basic types of contractions, isotonic or isometric contractions. In an isotonic contraction, the muscle changes in length as it moves the load, so that the tone of the muscle is going to change as it contracts. So this is an isotonic contraction in which the muscle is going to contract, and when it does that, it changes its shape, it becomes smaller, and produces enough of a force to lift a weight. So it produces enough tension to lift a weight. On the other hand, an isometric contraction, the word iso means same, and now metric meaning the shape. So in this case, the shape of the muscle stays the same. So there is contraction going on, there is tension being produced, but the tension produced is not enough to lift the weight. So the muscle remains with the same length. There is tension being produced, but the tension does not lift the weight. Examples of these would be lifting grocery bags or lifting an object will be an example of an isotonic contraction. Carrying grocery bags without lifting them would be examples of isometric contractions. So you should know the difference between isotonic versus isometric. The next question, the next portion is going to look at muscle metabolism, or the mechanisms, the mechanics by which muscle, skeletal muscle to be specific, creates ATP, creates energy. And when it comes to muscle metabolism, there are three basic mechanisms by which muscles, skeletal muscles, can make ATP. One of the mechanisms is called direct phosphorylation of ADP by creatine phosphate. The other one is called anaerobic pathways or glycolysis, and this is a fermentation pathway. And the last one is aerobic respiration. The very first pathway here, direct phosphorylation of ADP by creatine phosphate, is a very quick way, one-step mechanism to phosphorylate ADP and make ATP. So this is direct phosphorylation, a quick one-step process that makes ATP. This is unique to skeletal muscle cells, and actually cardiac muscle cells also have this mechanism. So it's unique to cardiac and skeletal muscle cells. It produces enough ATP to last for a few seconds. The process requires an enzyme called creatine phosphokinase, or CK. So creatine kinase or creatine phosphokinase, same enzyme, CK. What the enzyme is going to do is going to take ADP and a compound called creatine phosphate. Creatine phosphate is a phosphorylated carbon compound which is found inside a skeletal muscle. Creatine is made from proteins, actually. So creatine phosphate, ADP, plus creatine phosphate, an enzyme called creatine kinase or CK, sometimes also called creatine phosphokinase, is going to take the phosphate from creatine and transfer it and give it to ADP, adenine diphosphate. In the process, ADP acquires one more phosphate and it becomes ATP, adenine triphosphate, and this is the energy molecule. Creatine is now dephosphorylated and it's now it has lost its phosphate. It has been transferred to ADP. This is a very quick one-step process. If you remember, making ATP is a very complex multi-step process. This mechanism allows cells to create a little bit of ATP, about 15 seconds worth of ATP, very, very quickly in a one-step process. So this is one mechanism that skeletal muscle cells have to create ATP. Another mechanism, this one is again unique to skeletal muscles. In this mechanism, fermentation takes place. This is an anaerobic pathway. And what happens is when a muscle is creating tremendous amounts of force and it contracts at about 70% or more of its contractile activity, blood vessels are constricted and there is less oxygen, less blood flow, therefore less oxygen delivered to skeletal muscle cells. So bulging muscles are going to compress blood cells, oxygen delivery is impaired, so now the muscle cannot make ATP by aerobic respiration. When that happens, the muscle turns to fermentation. So let me show you what actually happens. So typically during aerobic respiration, glucose is taken into glycolysis and pyruvic acid is going to be produced along with a net total of 2 ATP molecules and 2 NADHs. So what glycolysis is going to do right here, glycolysis is going to oxidize glucose. Glycolysis oxidizes glucose into pyruvic acid, and in the process a little bit of ATP is made, but more importantly pyruvic acid is produced. Pyruvic acid is then taken into the mitochondria where aerobic respiration will take place. Okay. Now, during strenuous muscle activity that compresses the muscles, there is no oxygen available. So glycolysis still takes place because glycolysis doesn't require oxygen. But now, instead of taking the pyruvic acid into the mitochondria to make ATP, the pyruvic acid now stays in the cytoplasm and it's going to be fermented. And so let me show you what is going to be happening, or the purpose of all these. So you have glucose, and through the process of glycolysis, pyruvic acid is produced. The process makes a net of 2 ATPs, but the process also creates a net of 2 NADHs, so NAD plus is reduced to NADH. So now this NADH is going to be taken to the electron transport chain to deliver its electrons. However, under anaerobic conditions, when the muscles are being contracted so much that it prevents the delivery of oxygen, now instead of going to aerobic respiration, the muscle takes pyruvic acid and ferments it. Pyruvic acid becomes the molecule that is going to oxidize NADH and produce NAD again, so that glycolysis can continue and 2 ATPs can continue to be produced. In the process, pyruvic acid is going to be reduced to lactic acid. So the end product of fermentation in skeletal muscles is going to be acids, lactic acids to be specific. The process makes a net total of 2 ATP molecules per glucose molecule, which is not very much. As a matter of fact, it's not very much at all. Aerobic respiration would produce 34 ATPs. So this is enough ATP to last for about 16 seconds or slightly more. That's why strenuous exercise will tire us quicker, is because we're not going to have the energy. So this is the second mechanism by which skeletal muscle cells can create ATP. The third mechanism by which skeletal muscle cells create ATP is the most efficient mechanism, that is called aerobic respiration. So this gives plenty of energy for muscle contraction. So this will be aerobic respiration. Anaerobic respiration is going to make most of the ATP during muscle activity, during rest and during light moderate exercise. Aerobic exercise will also produce ATP by aerobic respiration. One of the advantages of aerobic respiration is that it's not dependent on glucose only to make ATP. Aerobic respiration can use glucose, but it can also use fatty acids or even amino acids, even proteins. So there is more of a variety of sources of energy with aerobic respiration. If the immediately available glucose runs out, no big deal. Skeletal muscle cells can actually store glucose in the form of glycogen, and this is unique. No other cell can do this except for the cells of the liver. So glycogen is a storage of glucose, which is found in skeletal muscles and liver cells. So let me show you the depiction of aerobic respiration. This is the aerobic pathway. Glucose is broken down in the cytoplasm to pyruvic acid. The pyruvic acid enters the mitochondria, and within the mitochondria, with the help of oxygen, which is going to go in, oxygen will be used to go through the electron transport chain. In the process, about 32 ATPs are going to be made. So this is a lot of ATP made per glucose molecule. The reactions also make CO2 and water. Also notice that aerobic respiration could use fatty acids as sources of energy or even amino acids from proteins. So it's not exclusively stuck with needing glucose, whereas fermentation needs only glucose, can only use glucose. And obviously creatine, the one-step phosphorylation of creatine phosphate will need just creatine or creatine phosphate. So this is next PowerPoint shows you a depiction of when do we use these different types of mechanisms to make ATP. So it says here that the first 6 seconds of a sprinter, for example, the ATP stored in muscle will be utilized first. Then the next 10 seconds, the ATP made is coming from the quick one-step creatine phosphate phosphorylation. Then after that, the next 30-40 seconds to a minute will be made by fermentation. By the glycogen storage in muscles is broken down and glucose is oxidized to generate ATP. This is typically during a sprint race. This will be done through anaerobic processes or fermentation. So that's another name for anaerobic processes, fermentation. The reason why is because during a short duration sprint type of exercise, muscles are being constricted a lot, trying to pick up speed. And there is, because of the level of muscle contraction, there is no oxygen available for the muscles, and they have to turn to fermentation. Now, during a prolonged duration exercise, like a marathon race, for example, the muscles can last for hours, because under these circumstances, there's plenty of oxygen being delivered, because the muscles are not being constricted in an strenuous manner. This is a more relaxed type of constriction, because it's an resistant type of exercise. So this is going to be the ATP made under those circumstances will be through aerobic respiration. So aerobic respiration is used during aerobic exercise and during at rest. So you should know the characteristics of these three mechanisms to make ATP. You should know what is glycogen, and what is creatine, and what is creatine kinase, also called creatine phosphokinase. Okay. Now, muscle fatigue itself is the physiological inability of the muscle to contract, and is typically due to imbalances in ions. Some of the ions needed for muscle contraction will be calcium, potassium, phosphate itself. Something else that happens during exercise, especially if it is a strenuous exercise, the sarcoplasmic reticulum is going to be damaged, and that's going to prevent the regulation of the release of calcium. Also, ATP may be depleted, even though that's not common. It could also happen. So there is a ionic imbalance, a loss of calcium, damage of the cells themselves, and eventually there may even be a lack of ATP. After exercise, there is an oxygen deficit that has to be repaid. So oxygen storage has to be replenished. Remember that oxygen in muscle, skeletal muscle, and cardiac muscle is stored in a molecule called myoglobin, which is unique to skeletal and cardiac muscle. So after exercise, oxygen reserves need to be replenished, glycogen storage need to be replenished, and ATP and creatine phosphate reserves have to be replenished. Now, during exercise, there's also heat production. Remember that about 40% of the energy released during muscle activity is used for work, which leaves us with 60% that is going to be used to give off heat. So, comparatively speaking, this is a pretty efficient mechanism. A car, for example, will be a lot less efficient than skeletal muscle is. The 60% given off as heat is what keeps the body temperature constant, and is also involved in thermoregulation when we're cold, for example. Okay, now notice that as we were talking about these mechanisms to make ATP, we mentioned several molecules that are unique to skeletal muscle, and some of them are actually also found in cardiac muscle, which give skeletal muscle its ability to contract for a long period of time. So one of them is that skeletal muscle, other than liver cells, are the only other cells in the body that store their own glycogen. Glycogen is a storage form of glucose. So skeletal muscle cells have stored glycogen to be broken down as glucose when needed. Creatine phosphokinase or creatine kinase is very special enzyme that can take creatine phosphate and donate the phosphate group to ADP to make ATP in a one-step process. Creatine phosphokinase is unique to skeletal muscle and cardiac muscle. And then finally, myoglobin, which is an oxygen storage molecule. And this one is present in cardiac and skeletal muscle. Smooth muscle is considered white muscle. Myoglobin is what gives skeletal muscle the red color. Myoglobin is similar to hemoglobin. Okay, So that is the end of the B set of PowerPoints. I'm going to go ahead and go back out, unless there's some question we can address. I'm going to go for PowerPoint C. Okay. So these next PowerPoints look more at how skeletal muscle functions. So force of muscle contraction, how strongly a muscle contracts, is dependent on the number of fibers that are going to be stimulated, and those can increase via recruitment of motor units. It's also dependent on the relative size of the fibers, and part of the principle of these is that skeletal muscle fibers are present in different sizes. So within a given muscle, there are different sizes of or fibers of skeletal muscle cells. Some are larger than others. During exercise, there is damage to the skeletal muscle. As that skeletal muscle repairs, it grows larger because in response to a greater demand that is being done on it, the muscle responds by making more myofibrils. By making more myofibrils, the muscle becomes larger. So the more we use a muscle, the more the muscle hypertrophies or becomes larger. So that is also going to, the hypertrophy of a muscle is also going to influence the force of the contraction. So what influences the force of the contraction is the number of muscle fibers recruited, the size of the muscle fibers stimulated, the frequency of the stimulation, and the length-tension relationship, which is what we're going to talk about next. Okay, so you should have an idea of the factors that can influence the force of muscle contraction. So if we want to increase contraction, we want to stimulate the large muscle fibers of a muscle. But to do that, we stimulate with high frequency of a stimulus, and we have to stimulate a large number of muscle fibers. Muscle and sarcomere will stretch to slightly over 100% to give us the maximum force of contraction. So when it comes to the stretch of the muscle, how much the muscle is being stretched or constricted during muscle activity, that will have a bearing on the force of contraction. This is what we call the length-tension relationship. Muscle, to give its maximum force of contraction, would have to exhibit between 80 and 120% of the contraction of the normal resting length. If we stretch a muscle past that 120% size from rest, the stretch will affect the zones of overlap, making it smaller and making the contraction weaker. If we contract the sarcomeres below 80% of the optimal range, now we're going to increase the overlap zone and decrease the number of cross bridges that can be formed, which means a decrease in the strength of contraction. This is a little bit of a difficult concept to visualize, is the relationship between the length of the muscle and the tension the muscle can produce. So here what we have is the sarcomere during a resting stage is between 80 and 120% contracted. Actually, I should say 100% contracted at rest. So at rest, the sarcomeres are about 100%. If we begin to stretch the sarcomere past that 100% to almost 120%, that sarcomere will still produce very, very strong contractions. By the same token, if we constrict the sarcomere to where it is about 80% constricted, you will still create enough contraction. However, if we stretch the sarcomere past the 120%, now we've stretched it so much that thin and thin fibers barely overlap, so there will be hardly any cross bridges form. So a low number of cross bridges are going to be made under those circumstances. On the other hand, if we constrict the muscle so much that now there is no H zone for the thin filament to be pulled in towards the middle, now again the force of contraction is going to decrease. So the force of contraction decreases if we constrict the sarcomere further than 100 and then 80%. And on the other side of the spectrum, the strength of muscle contraction will decrease if we stretch the sarcomere past 120%. Okay, so again, a little bit of a abstract concept, but it is important. What this is telling you is that overstretching a muscle or overconstricting constricting the muscle gives us less force than keeping the muscle within a certain limit of contraction. Okay, so and this is important for something like the reason why we need to bend our knees when we pick up an object is because if we don't do that, we're going to stretch our muscles past the 120% and we could harm them. So that's what we have our body in a good position before we do some kind of a strenuous work. Otherwise, we're going to take our sarcomeres beyond these normal optimum range. Okay. So again, we're going over how muscles work. Another concept is the velocity and the duration of the contraction. And that's going to be influenced by the load that the contraction is trying to lift, by the type of fiber that is being activated, and whether the muscle has a predominance of glycolytic fibers or not. The fibers that are glycolated are also called fatigable fibers, the ones that can get tired easily. But what I want to concentrate on is the type of muscle fiber. So in order to vary the velocity and duration of a contraction, we need to look at the muscle fiber type that is being activated, the load, whether it's very heavy or very light, and the ability to recruit more motor units when more force is needed. So we're going to talk about the different types of muscle fibers that are available. So in a skeletal muscle, not all of the cells are going to be the same. Some cells are going to be larger than others. And so the larger cells that use aerobic pathways to make ATP are called oxidative fibers. These are the fibers that are going to need oxygen in order to make ATP. On the other hand, the glycolytic pathways uses anaerobic glycolysis to make ATP. That is, it's not going to make as much ATP, and the ATP will come from fermentation. So this is going to be, glycolytic fibers are going to use anaerobic respiration or anaerobic glycolysis. Keep in mind, if you're going to use anaerobic glycolysis, that means that the only source of energy is going to be glycogen. If we use aerobic pathways, we can use glycogen, can use glucose, fatty acids. So it has more of more choices, and amino acids, and of course glycogen, because it's essentially glucose that needs to be broken down. Okay. So we have three types of muscle fibers that we find in skeletal muscle. The ones we call slow oxidative fibers, the ones that are called fast glycolytic pathways, and somewhere in the middle are the fast oxidative fibers. So we have a slow oxidative, fast glycolytic, and fast oxidative. This table has the characteristics of all three fibers. We're going to concentrate on the two extremes, the slow oxidative fibers and fast glycolytic fibers. The ones in the middle kind of have characteristics of both. So now, main thing here is students have two options at this point. They either memorize the characteristics of slow oxidative and fast glycolytic pathways, or they can understand the differences between them, and from that understanding come up with the characteristics of a slow oxidative or fast glycolytic. So I think that the second option is a little bit better, especially if you don't have, if you don't have a good memory, like most of us don't have. So we prefer understanding than trying to memorize. So this is the deal. If you look at this picture right here, you see this smaller, darker fiber is a slow oxidative fiber. This larger, lighter color fiber is a fast glycolytic fiber. Fast oxidative here is the middle guy that we're not going to talk about. We're going to concentrate on the two extremes. So the first thing that you ask yourself is, which fiber is larger? Well, hopefully the obvious answer, oops, didn't mean to do that. The obvious answer is going to be the fast glycolytic fiber is larger. And what comes from that is, based on your answer from question 1, which one is the larger fiber? Which fiber will have more myofibrils? Because the larger the fiber, the larger the cell, the more myofibrils it can have. Well, if fast glycolytic fibers are the larger ones, then they are the ones that are going to have more myofibrils. So they're going to have more myofibrils. Okay, what comes from that is, if the cell has more myofibrils, that means a stronger contraction. The more myofibrils, the more cross bridges, the stronger the contraction. So based on your answer to question 2, which fiber will produce a stronger quick contraction? Well, fast glycolytic. So fast glycolytic pathways are needed when we need quick, fast, strong contractions. Okay, so strong contraction happens very quickly, then we use in the fast glycolytic fibers. Okay, next question is, which fiber is darker in color? Obvious answer, hopefully, is the slow oxidative fiber. So based on that answer, which fiber will have more myoglobin? The darker the fiber, the redder the fiber, the more myoglobin it has. Myoglobin is the storage molecule that stores oxygen. Very similar to hemoglobin that makes red blood cells red. In this case, myoglobin makes muscle red. So slow oxidative fibers have more myoglobin, which means it has, oops, I didn't mean to do that, means it has more oxygen storage. So the slow oxidative have more oxygen stored. Makes sense, because these are the fibers that are going to use aerobic respiration as their mode, as their metabolic pathway to make ATP. So these fibers are going to have more myoglobin. That means that they have more oxygen reserves. That means they're going to be fatigue resistant, because they're going to use aerobic respiration to make ATP. They're going to use oxygen to make ATP, which makes them fatigue resistant. They can't produce more ATP and work for a longer period of time. So based on the answer to question 3, which fiber will fatigue quicker? Well, the answer to that would be the fast glycolytic fiber, which is not using oxygen, is using fermentation, which means it's going to make just a little bit of ATP and it will not be able to work for a longer period of time. So this is what this leads us to. What this leads us to is that fast glycolytic fibers are going to give us a stronger contraction. However, so a stronger contraction of short duration. Slow oxidative fibers are going to give us a slower, weaker contraction because the fibers are smaller, but it's going to be fatigue resistant. So this will be fatigue resistant fiber. So we're going to use, this will be a fatigue resistant. weaker contraction. Okay, let me give you an example of when we use these fibers. A marathon runner will have lots of slow oxidative fibers and in his lower extremities. These middle ground fibers could be, through training, changed into slow oxidative or fast glycolytic. So someone who does a lot of aerobic exercising, their muscle cells will turn, more of their muscle cells will turn into slow oxidative fibers. On the other hand, a weightlifter, for example, will train its fibers into becoming fast glycolytic fibers. Another analogy is that of the kinds of muscles that we have in chickens. So white meat in chickens is found in the breast muscle. That is your fast glycolytic fiber. A chicken doesn't fly all day. A chicken only uses its wings when it's trying to run or fly away from a predator. So it needs the muscles of the breast to be very strong and produce very strong contractions very quickly to be able to very quickly fly away from a predator. On the other hand, a chicken does stay on its feet all day, walking around. So that's where we have the dark meat of the chicken, the redder meat. That's the slow oxidative fibers, which are fatigue resistant and allows the chicken to be on its feet, walking around all day. So that's another analogy. So yes, you do need to know the characteristics of these fibers and be able to compare and contrast them. And for that, again, we have these table right here. So you can see that these low oxidatives are smaller, fast glycolytics are faster. That is because the slow oxidatives can use aerobic respiration, so they make a lot more ATP, versus the fast glycolytic, which have to use fermentation, which makes less ATP. It says myosin ATPase activity. So myosin, well, we didn't really talk about that, but this is slow myosin versus the fast myosin. The primary pathway for ATP synthesis, aerobic versus fermentation. Myoglobin content, very high in the dark, slow oxidative fibers, low in the lighter, fast glycolytic pathways. Glycogen storage. Okay, slow oxidatives have a low glycogen storage, and that makes sense because aerobic respiration can use other things. It doesn't have to use glycogen. It can use fats. It can use proteins, versus fermentation, which only uses glycogen. So it has to have a high glycogen storage. Rate of fatigue, slow fatigue fibers versus fibers that fatigue quickly. Color very red versus white. Fiber diameter small versus large. Mitochondria many, because the mitochondria are needed for aerobic respiration, versus few because we don't have as many as being used for fermentation. Capillaries many because it needs the oxygen for aerobic respiration, and fewer capillaries on the other side. The slow oxidative are adapted for endurance activities. The fast glycolytics are adapted for short-term intense power exercise like weightlifting. So hopefully this helps to remember what is what, and if you just remember the picture, hopefully that can lead you to all of the characteristics of each of these two fibers. Okay. Let's see. Just wrapping up these, load and speed and duration of contraction. This is pretty self-explanatory. The lighter the load, the longer the contraction can be sustained, and the quicker the contraction can be produced. The heavier the load, the shorter the duration of the contraction, and actually this is the distance that the load can be lifted. So a lighter load can be lifted for a longer distance, versus a heavier load can be lifted for a shorter distance. And recruitment. Okay, so the concept here is that when we're trying to lift an object, as we stimulate the muscle in order to lift the object, is larger and larger motor units will be stimulated. So you're trying to lift a table and the first stimulation doesn't do anything, then you increase the stimulation and that's going to recruit larger motor units. That doesn't do the job, then you increase further and you continue to keep stimulating the muscle, and as you do that, larger and larger motor units will be recruited until the object is lifted. Okay. Which makes sense. You don't want to be stimulating the large motor units from the beginning, otherwise you're going to produce too much force. Effects of exercise on muscle. Aerobic exercise is going to increase muscle capillaries because aerobic exercise needs more oxygen. The number of mitochondria because it, again, needs more oxygen. Actually, because the mitochondria is the place where aerobic respiration takes place. And the synthesis of myoglobin because myoglobin will be the storage molecule for oxygen. So greater aerobic exercise will result in greater endurance, strength, and resistance to fatigue. And some of the fast glycolytic fibers may be converted to fast oxidative fibers in the process. Effects of resistant exercise, on the other hand, resistant exercise will increase the size of the muscle. Again, now you're getting the bigger fast glycolytic pathways. We're going to have, again, mitochondria will still be increased because not as much as on the other kinds of fibers, fast oxidative, but mitochondria is always going to be needed. So we're going to increase the number of mitochondria. The number of myofilaments will increase. That's part of the reason why the fiber becomes larger. Glycogen stores will increase because this type of activity requires glycogen. And connective tissue will increase also because these larger fibers will need more connective tissue. Skeletal muscle hypertrophies, meaning that in response to resistant exercise, the muscle becomes bigger. And just keep in mind that hypertrophy means that the muscle cells become bigger, not that we're increasing the number of muscle fibers. So forcing a muscle to work hard promotes an increased muscle strength and endurance. However, the muscles will adapt. So if you want to maintain or increase the strength of the muscle, you have to increase the demands you make on the muscle. Okay. So the last part of this chapter is going to look over the different types of muscles. Yet again, we kind of look at this in chapter 4. Now we're revisiting all these concepts and paying a little bit more attention to the characteristics of the 3 different kinds of muscles. So a smooth muscle, you know that it's found in the walls of hollow organs. There's usually many layers, more than one layer, and the layers are going in different directions. So there might be a circular layer and a longitudinal layer going in at 190-degree angles. A smooth muscle, the fibers are small, spindle-shaped. They have less connective tissue. They only have endomysium. The sarcoplasmic reticulum is less developed. It doesn't store calcium. Instead, the calcium is trapped at the surface of the muscle cell in little infoldings called caveola that are going to sequester or trap calcium outside the cell. There are no sarcomeres, no myofibrils, no T-tubules. This is non-striated muscle. Okay, so that's what's called a smooth. It is innervated by the autonomic nervous system as opposed to by somatic motor neurons. So it is an involuntary muscle, and that is just showing you the innervation of the autonomic motor neurons. Let's see, the myofilaments in a smooth muscle, they are not organized into sarcomeres. There is no troponin. Instead, the protein that binds calcium is called calmodulin. You definitely need to know that. No sarcomeres, hence no striations. One of the characteristics of smooth muscle fibers, since they don't have sarcomeres, is that when they constrict, they're going to constrict at at different angles. So it's not going to be just a shortening of the muscle, but it's more of a corkscrew type of constriction. And that's because the myofiber, the thick and thin filaments are going around at different angles in the muscle cell. They're not just around the longitudinal part of the muscle cell. So that's one characteristic. This is a muscle that is special because it can constrict even when it is stretched, which is something that a skeletal muscle cannot do. If you stretch a skeletal muscle, it cannot contract, because if you stretch it, you stretch the sarcomeres. A smooth muscle can continue to contract even if you stretch it, which makes sense because that is exactly what it's supposed to do. It is found in the stomach. As we eat and we stretch the stomach, the smooth muscle in the stomach is still supposed to be able to contract even though we're stretching it. So that's typical of a smooth muscle. Another characteristic of a smooth muscle is that it exhibits hyperplasia, meaning that when it is stimulated by hormones, for example, instead of increasing the size of the cells, the number of the cells is increased. So they can divide and increase in numbers, especially in response to hormones like estrogen, for example. Cardiac muscle, on the other hand, is found in the walls of the heart, making up the myocardium. It is also innervated by autonomic motor neurons. It has striations, it has sarcomeres, which tells us that the physiology of muscle contraction is going to be similar to that of a skeletal muscle. It does not have triads. Triads are unique to skeletal muscle. It has intercalated discs, which is unique to cardiac muscle. Intercalated discs are gaps that connect each cardiac muscle cells to each other. The cells are branched, uninucleated, and the response to repeated stimulus is hypertrophy, not hyperplasia. So in that regard, is similar to skeletal muscle, meaning that if we force the heart to work and constrict more, as it may happen if we have high blood pressure, for example, the muscle will become bigger and bigger and bigger. And that's a depiction of all three types of muscle. And again, it goes over the characteristics of muscles. The effects of aging in muscles. As we age, muscle loses its mass. That is called sarcopenia, the loss of mass muscle. Sarcopenia could be reversed through exercise. And then the last feature talks a little bit about muscular dystrophy, which is a genetic inherited abnormality of the muscle, in which there is a protein lacking that stabilizes myofibrils and sarcomeres. So if myofibrils and sarcomeres are not stabilized by this protein called dystrophy, the muscle begins to break down, and that happens over time. It causes atrophy of the muscle. It's a sex-linked. This muscular dystrophy is a sex-linked condition, genetic condition. It's found mostly in males, and it's a degenerative condition in which the muscle progressively becomes weaker and is not able to function. Okay. What I want to do before I close is I want to get out of here really quick and summarize again. Just a second. Get the whiteboard and summarize again the characteristics of the different types of muscles. So what we did is we had skeletal muscle. Okay, we had a skeletal muscle, cardiac muscle, and a smooth muscle. And we talked about the characteristics of these different types of muscles by looking at things like location where the muscle is found. So a skeletal muscle will be found attached to bones and skin. Cardiac muscle is found in the walls of heart. A smooth muscle is found in the walls of viscera. So the location is different for each one. Another characteristic we talked about was striations. And in that regard, skeletal muscle had striations, so did cardiac muscle, and there were no striations for smooth muscle. With the striations came sarcomeres. Okay, so if the muscle has striations, then it has sarcomeres. If it has striations, if it didn't have striations, it doesn't have sarcomeres. Okay. Another characteristic we talked about was the protein that binds calcium. So calcium binding protein. Okay. In the case of a skeletal muscle, the protein is troponin. In the case of cardiac muscle, the protein is troponin again. In the case of a smooth muscle, is calmodulin. Triads are found in a skeletal muscle, and that's basically it. Cardiac muscle also had intercalated discs. That's unique. The fibers were branched in cardiac muscle, and one nucleus. They were spindle-shaped in a smooth muscle, and one nucleus were long rods, very long. These are the biggest fibers. So long rod-shaped fiber in a skeletal muscle, and they were multinucleated. Another characteristic that we talked about was whether the muscle was voluntary or involuntary. Skeletal muscle was voluntary, which doesn't mean that we're always going to use it voluntarily. It just means that we can if we want to. Cardiac muscle was involuntary, and smooth muscle was involuntary. So with voluntary and involuntary came also the type of motor neuron that activated the muscle or innervated the muscle. In the case of a skeletal muscle, running out of room here, skeletal muscle used somatic motor neurons. Cardiac muscle and a smooth muscle both use autonomic neurons, or I should say actually autonomic motor neurons to get a little bit more precise. Okay, I think those are all the characteristics that we talked about that I can think of off the top of my head right now. So be prepared to, again, compare and contrast these three different types of muscles based on their characteristics. Okay. Just to go back, when it comes to the physiology of skeletal muscle contraction, go back to the sliding filament theory of muscle contraction. Be very helpful to watch the videos. Remember that the sliding filament theory of muscle contraction had three parts to it: the event at the neuromuscular junction, the excitation, contraction, coupling, and cross-bridge formation. Be able to recognize the events and match them with their names, what happens at each event. During events at the neuromuscular junction, the somatic motor neuron released the neurotransmitter. Definitely know the neurotransmitter released by the somatic motor neuron. If this is the little neuron there, and let me make the little muscle cell here. That's the muscle cell, muscle fiber. The neurotransmitter released, I'm going to make it in different color here, purple. Okay, the neurotransmitter released, which is released from vesicles inside the somatic motor neuron, is acetylcholine. This is not going to go away. It's going to come back on next chapters. The acronym is ACH. Acetylcholine binds to acetylcholine receptors found in the cell membrane of the muscle cell. The cell membrane is called the sarcolemma. And the receptors are going to bind acetylcholine. And when that happens, the receptor, which is an ion gate, allows for sodium to go inside the cell. Sodium is positively charged. Under normal resting circumstances, the skeletal muscle fiber is negatively charged. That is the resting membrane potential. When the muscle cell is stimulated and sodium goes in, now the cell is going to become positive. As a matter of fact, it's going to become very positive, go as high as 30 millivolts, positive 30 millivolts. That's what we call depolarization. Sodium is involved in depolarization. That is the signal that depolarization, those sodium ions, travel inside into the T tubules. And now we are on the second step, which was the excitation-contraction coupling. The sodium ions traveling inside the cell and making that cell more positive opened the storage of calcium inside the sarcoplasmic reticulum. I'm going to make calcium, I guess, black. All right, so calcium is going to be black. So that's a little calcium inside. These gates open when the sodium goes inside the T tubules. Now calcium is outside the cell in the cytoplasm of the cell, available for troponin to bind, and then we have the cross bridge formation. So looking at a description of the event, you should match the description with the name of the event. You should also have an idea of what the role of calcium is in muscle contraction. Remember, calcium is going to be needed to release the neurotransmitter. Calcium needs to go inside the neuron for the neurotransmitter to be released, which means that we have calcium outside the muscle cell. We also have calcium stored inside the muscle cell. Calcium is going to be needed to be released inside the muscle cell from the storage in the sarcoplasmic reticulum into the cytoplasm to initiate the formation of a cross bridge. The other question would be, could be, what is ATP? What is the role of ATP? The role of ATP will be to break the cross bridges. Formation of cross bridges does not require ATP. What requires ATP is the breaking of the cross bridges in order to relax the muscle. This is counterintuitive. We may think that muscle contraction requires ATP. That's not it. Muscle relaxation requires ATP. The contraction doesn't require ATP. Contraction is automatic formation of the cross bridge between myosin and actin. That explains rigor mortis. That explains the contraction of muscles when we die. In a corpse, the minute that death happens, the cross bridges remain because ATP is gone from the cells. The cells are not making any more ATP. Therefore, cross bridges cannot be broken down and muscles remain contracted. And that's what causes that muscle contraction after death. Is the lack of ATP to cause muscle relaxation. So those are the kinds of questions that you might find in the test. Is the role of ATP, the role of calcium? What is the neurotransmitter? What is the name of the neurons? It's called a somatic motor neuron. Okay, hopefully that helps. Any questions? Okay, so I'm gonna sign off. And we will continue to schedule sessions. Oh, yeah. Oh, good question. Somatic. Oof. Okay. Yeah, the problem with somatics that we're not really going to talk about it until the next chapter, but since you ask, somatic means body. And the neurons that innervate skeletal muscle are called somatic versus visceral. because we tend to, in order to move the body, we need a skeletal muscle. And that's what they're called somatic, is because of body. Somatic means body. So these somatic motor means movement. So somatic motor neurons are the ones that innervate a skeletal muscle, which means they're the ones that move the body, versus the visceral motor neurons, which are the autonomic neurons that innervate viscera and the heart. That's not considered the body in the sense that that's internal. Okay, so that's why the name somatic comes in. It means body. And we'll use it again in chapter 11 to describe other types of neurons. But hopefully that explains that part. Maybe, yes. Okay, yeah, you're welcome. Okay, any other questions? Okay, very good. So I'm going to sign off. Do you have another question? No, okay. So I'm going to go ahead and sign off. Okay, and I will load the recording as soon as it gets ready. Okay, have a good evening.